season is coming. So we have for you in Olav as well. I forgot that. Okay, so our next speaker is Olav. He's leading the Digital project. He's from the Norwegian University of Life Science, and
thank you. And the microphone seems to be okay. So I feel very strongly that the powerful methods that we have heard Evan describe and that Bernard has developed are ripe for being applied in food production. So I will talk about systems biology for salmon farming, and I also believe that this is the direction that all of the various food production species and plants will eventually go. But salmon has been a nice place to start for Norway, having recently sequenced the genome and it being Norway's second biggest export industry after oil. So I will look a little bit about metabolic modeling because it is where we chose to start, but I will also talk a bit about what lies beyond metabolism. Especially in human biomedicine, there is a lot of other systems biology also being developed, which transfers nicely when we try to go from the single cell to the functioning multicellular organism. So this is hairier in many ways than working with bacteria, and thus there are more limitations on what we can do, but I believe it will be very fruitful to try. So the farming of salmon in Norway generates meat that is being sold all over the world and generates a huge amount of money. But this growth in production has brought many challenges. One of them is how you're going to feed the farmed fish when there isn't enough bycatch and leftover fish pieces to feed the salmon with. And another one is then to try and ensure that the salmon fillet is equally nutritious and tasty and looks as good as for the original wild population. There are many challenges with disease when you keep the animals very densely together, illustrated by sea lice here. And it is also a challenge to preserve the wild populations that this bonanza came from. So what these challenges have in common is that the solution to them will involve understanding complex interplays in living things. And this is what brings us to systems biology, where we view the functioning living thing as a set of components or organs, if you like, that affect each other and depend on each other. So, just to illustrate a bit about the value of formulating mathematical models, consider this stylized creature which looks a bit like a turtle or a baby. And it is described through parameters. How long are the forelegs, the hind legs, what's the angle of the neck? But then if you raise the head a bit and elongate the neck, you get something that looks a bit like a llama. And if you elongate it further and have it bow a bit, you get a fair approximation of a giraffe. So in making this caricature, we have done two things. We have shown for a whole class of systems what they have in common, namely the form of the equations or the schema that we used to build them. But we also show how they differ, namely in the actual values of the parameters. So constructing the model both distills the essence of this class of organisms and gives us a relevant basis of comparison, mathematically speaking. So, this will be my text-heaviest slide, I think, I promise. The ambition for the Digital Salmon is to build a library of mathematical models that can serve as components in putting together models for different aspects of salmon life. So it will take the form of mathematical equations and computer programs that encode biological knowledge. And they can then be adjusted to represent different states of the salmon, whether it is growing fast, growing slow, whether it is healthy, whether it is sick. You can use it to represent different stocks of salmon, if you have local adaptations in different rivers. And I'm sure you can use it to compare it with neighboring species, such as trout. And this will have application in answering the challenges that I outlined on the previous slide. And I don't think we will do this within a 4-year project. It's going to be a research program for decades, but I think it's a very necessary investment in the organism that produces this value for us. Unfortunately, clever people have done a lot of the groundwork for us. So this is Bernhard's book, and this is an illustration of a single-cell organism, which is where it is often pretty obvious that fitness equals growth rate. This becomes more complicated when you look at a higher organism where you have organs. So the liver is not made to grow, it is made to serve a purpose within the context of the body. But there is a very detailed metabolic reconstruction that has been made for humans, and it can't be much more difficult with salmon. But it's interesting to see that this is what Bernhard means by systems biology, because there are also other traditions that can validly be called systems biology. And the virtual physiological human is an attempt to make models of the function of the major organs, and the flagship example is the heart, which is an electrically controlled mechanical pump. It requires entirely different equations to describe the essence of what it does than when you look at the metabolic reaction network. And we'll get back to this in one more slide, but this is a project that has been in the inception since the 70s, but is beginning to gain traction with the availability of omics data, but also imaging data. So this illustrates how the heart and the lungs and the skeleton and the liver all have their separate sub-projects that try to describe mathematically the function of the organs, meaning what is their input, what is their output, and what are the mechanisms that make up this function. It spans scales from the very small to the very big, and from the very fast to the very slow. And it is a huge challenge to combine across scales the quite detailed and in-depth knowledge we have of each of these phenomena. Yes, this is also an interesting point, that we often use models that are not mathematical but in the lab. So having an isolated cell can illustrate what the cell would do in the context of a tissue. Having a fish in a laboratory can illustrate what it would do in a net cage. So, using the heart as an example again, we have the whole range from genes to proteins, and then the structure of the proteins determines their function in the cell, which can be stylized mathematically like this. And then you get, as the main output, the propagation of an electric signal and contraction of muscle proteins in response to that signal. This gives you parameters in a tissue description. And if you also have the stiffness of the heart tissue and the direction of muscle fibers, you can begin to simulate the function of the entire heart. And a very important part of the groundwork that has been laid by the Physium project and also by metabolic modeling is agreeing on data formats and agreeing on ways of labeling the variables in the equations so that the interface of one model matches the interface of another. So we are a partner in what is called FADOM, which is a consortium to make data and model resources findable, accessible, interoperable, and then reusable. So what this enables is that when we get new data or we develop a new model, it can automatically, or with just a little help, connect to what we already understand. And it makes it easier for us to quickly survey what we know, what data do we have, what processes have been modeled, and where are the knowledge gaps. So back to the salmon, there are many, many challenges, there are many phenomena, and where should we begin? And this is a thing we thought long and hard about. The answer is you want to start with something that you can both manipulate, measure, and model. Because if you're missing any of these three, you are in for trouble. So in our case, which I will describe in more detail shortly, we look at the challenges of sustainable feed for salmon. It is something you can experiment with in aquariums, feeding the fish different stuff. You can mimic this in the lab using small pieces of tissue, and you can collect relevant data using gene expression measurements and metabolomics. And it matches very well the mathematical framework that Eivind outlined in the previous talk. So then, starting with this sustainable food production, the thing is that salmon is a carnivore. It eats other animals in the wild. So when we first started farming salmon, we gave it something like this to eat. But then, as aquaculture increases, most of the fish oil and the fish meal is consumed by aquaculture, and still it needs more. So then we taught salmon to eat this. And today, 3/4 of both the fats and the proteins in a typical fish feed comes from plants. And this has been a long and arduous process to find out how you can compose a diet and counteract the tendency for this very unnatural food to give inflammation in the gut, and you can still keep the fish growing well and producing good meat. So now researchers are looking into alternatives to these plants, because the fields where you grow the soybeans, you could use to produce plants for human food. So even though there is more available than the fish meal and fish oil, it is not necessarily more sustainable. So then this lady and something called Foods of Norway is an industry-heavy research project that is looking into sustainable alternatives using feedstuff made from yeast or made from sawdust, made from seaweed, that kind of thing. And this poses a pretty complicated optimization problem, because you have many tens, if not hundreds, of potential ingredients, each with their own availabilities and prices, which fluctuate rapidly, and each with their own effects on the organism. So you want to quickly be able to compose a diet that provides good meat, which is not too expensive, which is sustainable, and which keeps the fish healthy. And I feel strongly that that requires us to get a systems understanding of the salmon body. So, aquarium experiments are simple. You feed the fish, you wait a few months, you pay 500,000 kroner, and you make the measurements. But the cheaper and faster alternative is to do tissue slice culture. So this is just a few colorful photographs of how you do it on liver and fish. You cut the fish, take out the liver, cut it into little pieces, which you literally super glue onto a plastic cylinder and encase it in agar and watch it congeal. This you then mount in a vibratome, which has an ordinary Gillette razor blade that vibrates and cuts off thin slices, you can barely make one out, which you then put into their own Petri dish, and you can then in vitro feed them different things. In this case, vegetable oil versus marine oil versus a medium with no fat source at all. And then measuring, this is just a generic illustration of the fact that you can measure the expression of thousands of genes at a time. And you can also measure thousands and hundreds of molecules, the abundance of them, using metabolomics, even though it's slightly more complicated than I imagined when I started. And the good thing about this is that it fits right in here. Anybody who has had high school chemistry will recognize the formulas, and you see how the molecules get rearranged and changed via the help of enzymes. Most of these reactions are catalyzed by enzymes, which are coded for in the genome, and which are expressed under some circumstances and maybe not under others. So by taking a combination of RNA expression and metabolomics, you get measurements both of the abundance of the molecules and of the abundance of the enzymes. So it's a very nice match between what we can manipulate, measure, and model, and it is related to a pressing challenge in salmon farming. And this is why we chose to start here. Yeah, Eivind has said this very nicely. And it's also a nice starting point in that you can use the same framework as Bernard has explained in his book to cover signaling networks, where it is not so much the gross transfer of matter that is the point, but the transfer of information. So signaling networks helps the apparatus of life sense what is going on outside and send signals to the other parts of the organism. It ties in with energy metabolism, and it ties in with ion transport, and thus to the heart modeling that we have been looking at. It ties in with osmoregulation, which is important to salmon, because as most of you know, it lives in fresh water when it is born, and then it migrates to salt water and then back again. And this requires readjustments of the metabolic network that has impact on the energy in metabolism. And as I said at the beginning, I think this is where production biology is headed. And I think that cattle and wheat and anything is going to follow suit. I'm not sure how long it will take. But I think that it won't be very long before every biologist, in some sense, is a systems biologist. They may not be mathematicians, but I think it will be commonplace that they are good users of models. They can be critical of the assumptions of models, and they can interpret the output of them and can contribute to their formulation. And this is not saying we should not be doing big data and machine learning. It is a complement to what you can do with statistics and the current buzzwords, to look for patterns in big data, and then you can talk to a biologist, you can do experiments in the lab, and try and connect it to physiology and to mechanism. And eventually, I foresee that there will be businesses to be built on top of this. A bit similar to how you make the weather forecast. You could be providing suit-tailored recommendations on how you should be feeding salmon under given temperatures or for a given strain of salmon. And the key thing is that you have the user's own data to put into this, so it is personalized, if you like. Also, there will be a steady stream of new feedstuffs becoming available or being considered. And you will then have a system where you can rapidly screen in silico and with a strategic selection of experiments, find out what are the effects of this feedstuff, and then simulate what are the likely repercussions in the body. And this helps speed up the process, it helps save a lot of research animals, and it helps save cost, because you get a much better and more plausible selection of hypotheses that you try out in real life. And this change will be facilitated as we get more and more automated data collection techniques. Omics is nice, but there is also a lot being done in video surveillance of fish, studying how they behave, for instance, and imaging, 3D scanning, that kind of thing. So it helps to feed the data-hungry models. So as the models get more complicated, we also get more data. And this is something that is motivated by industry and money. We will build a knowledge base for salmon that we could never have gotten funded for as basic research in ecology and management. But it will be useful for ecology and management. There will be opportunities to contrast the farmed salmon with the wild salmon and with other species that will, with a little strategic sampling and a few strategic experiments, you can take your new species and you plot them in the space of model parameters. Yes, and that will have to serve as a punchline. This is a very transdisciplinary project, and most of what I have been describing now is the long-term vision. So it's a long way ahead, but I think it's very worthwhile to start walking it. Thank you.
Thanks a lot, Olav. We have time for questions.
To speak up so you can hear me. I mean, this is a very kind of simple-minded question. That if you're not going to feed salmon soybean, do you have any ideas what the alternatives could be?
Yeah, one of them was basically sawdust. I have been told not to call it sawdust, but something nicer.
Sawdust.
But the idea is you have polysaccharides in what is left over from forestry. And with the right enzymes, you can break up some strategic chemical bonds, and then other bacteria can get at this, and yeast will eat the bacteria, and the yeast becomes a protein source that you can include as a foodstuff. And I think people are looking into algae, and overall getting things from the ocean is kind of the standard answer to this. I know people are looking into harvesting what they call mesopelagic fishes, the ones that are deep in the ocean and not normally harvested. Yeah. We could imagine joining forces on doing something similar for cod, if you like.
Any other question?
Yeah. It's just a great talk. Very enjoyable to listen to. I was reminded of two or three things as you spoke. First is that the very original review of cross-balance analysis from 1984 showed that they're actually good for nutritional purposes. They can predict feeds and responses of organisms to, mostly microorganisms to nutrients. There are some papers coming out now that study how the worm C. elegans is fed with different bacteria, different strains of E. coli. And depending on which strains you feed it, it responds differently to blood, for instance. So when we start using FDA for higher animals or complex animals, there'll be a lot of interesting biological issues that will come up. But I think the FDA type of approach will probably help quite a bit here. The other thing I wanted to say is that you pointed out this name systems biology. I just want to say a little bit about its origin. It really should be molecular systems biology, because before it came on the scene, a lot of systems biology existed in different parts of biology, like in ecology and immunology and so forth. But people were so excited around 2000 that they dropped the word molecular. So it really should be, the title of that book should really be molecular systems biology, as I think it says in the preface. But for some reason, the word molecular just disappeared early on. They claimed the whole field for themselves. So apologies for that.
Yes, I have lots more to discuss, but
Okay, so thank you. Let's thank the speaker again.